Hi everyone, welcome to RPI Data Science. So from today on, I would like to talk about meta-analysis in R. So from the very basics of meta-analysis, we will dive to more complex topics in meta-analysis, and we will cover them in the next series of videos. But before we go much deeper in meta-analysis, I would like to give you some useful references which I will also use in my next videos. First one is this meta-analysis in R book, meta-analysis with R. This is really a very nicely written book by Guido Schwarzer and his colleagues. This is probably, if you have access to Springer books, you can get a free copy. Otherwise, it's also available in Amazon or something. The other one that I would like to recommend is really a very nicely written hands-on guide, meta-analysis in R. This is also freely available on the internet. You can access it. This is really one of the nice, especially if you are going to employ R for your meta-analysis. This is really nice and a very recent book which is written using R actually. The third reference, of course, that I would like to show is this the Cochrane handbook. You know, every meta-analysis or every systematic review and meta-analysis follows the Cochrane guideline. So it's useful. I mean, it's very important to be familiar with handbook, because if you are doing systematic review and meta-analysis, you will often have to refer this guideline, I mean this handbook, very frequently, which is written by Professor Julian Hicks. So these are the three main books that I very much recommend, and here are the outlines that I will probably cover in the next series of videos. We'll talk about effect sizes and how to pull effect sizes using the standard methods, and we will also talk about heterogeneity and visualization aspects, how you visualize in terms of forest plots. And we will also talk about subgroup analysis, sensitivity analysis, meta-regression, publication bias, and probably in more complex topics like network meta-analysis and the like. But some of the topics that I would like to focus are really very basic. Maybe we may not dive very deep into the more statistical nature of all these meta-analysis topics, but we will cover most of the necessary steps. I mean, the necessary topics, at least, that you will employ in your meta-analysis. So to use, I recommend a prerequisite to have some basic R experience and some epidemiology and statistics. These are really very important prerequisite knowledge that you would employ if you are going to do meta-analysis in R. Then there are many packages. There are so many packages actually, probably about 60 or I don't know, more than 50 or something. There is actually a paper on the review of meta-analysis packages in R. You may find it on the internet, and you can check it out. But these are really some of the most popular R packages that you may come across. But I am mainly focusing on meta and metaphor, because some of the packages, like in some of the functions that meta uses, you may have to also load from metaphor. So it's very important to install these two packages, and I will give you another package which is ESC package. So these are really the basic package, the most important package actually that you will need for our tutorial. Then after that, let me give you a brief overview of how meta-analysis evolved over time since its introduction in the late 1960s and up to now. You see, this is actually my most recent search in PubMed. I was simply searching meta-analysis and the term meta-analysis in PubMed, and here are the number of publications that were published across time. So there is really an exponential growth of publications that contain the term meta-analysis. Actually, meta-analysis is one of the most influential research evidence to influence clinical, public health, or economics decision making. So meta-analysis is actually a very important field of research. So you see how it has been increasing over time. So it started in the early 1960s, and then it goes it exploded in like where we are now, 2020s or something. So it's ever increasing. So that means there is huge interest in this field. You may also be conducting meta-analysis, or you might have already conducted meta-analysis, and so when you talk about meta-analysis, the first thing that comes to your mind is effect sizes, because you want to synthesize effect sizes, and we'll talk about effect sizes. And what are effect sizes? Effect sizes are actually a quantitative measures of before we define effect sizes, let's talk a little bit about meta-analysis. And meta-analysis is a technique which actually summarizes effect sizes. You can have multiple, different effect sizes from different reporters, and based on their nature of similarity or comparability, you will combine this. So meta-analysis gives you a technique to summarize or to synthesize those measures. So effect size is usually mostly denoted by theta, this Greek letter theta, and it's usually a metric to identify or to quantify, and it could be relationships, or it could be something that to quantify something. It could be mean, or odds ratio or risk ratio may quantify something. At the same time, it may also quantify relationships. So that's actually called effect size. So effect size captures the direction and magnitude of relationships, and examples of there are many different examples of effect sizes. So you may have mean, proportion, correlation, risk ratio, odds ratio, hazard ratio, incidence rate ratio, or something. There are many different effect size measures that you may come across when you deal with meta-analysis, or when you deal with some statistical analysis to quantify something. And so effect sizes has to be, in order to to combine them or to use them in meta-analysis, they have to be comparable. And that means they have to have the same measurement, or at least comparatively, comparable. So that means they have to have similar meaning across studies. So if you talk about depression, the depression that's measured in one study has to use the same scale with the other one, so that you will be able to synthesize them, or you'll be able to combine them. Otherwise, you will have to find out a technique to standardize this depression that's measured with different scales. And the other important characteristics of effect size, when you have to combine them in meta-analysis, and it has to be computable. That means you have to derive them numerically. You have to be able to calculate. And reliable. So that means if you measure using that effect size, if you measure it multiple times, there has to be some kind of standard error, or deviation from one measure to the other should be able to be measured. Means it has some standard error. And then another important characteristics of effect sizes that you need when you combine them in meta-analysis is it should be interpretable. It should be meaningful. When you talk about mean or odds ratio, what does it mean? So it has to have some kind of meaning attached to it. So these are some of really the most important characteristics of effect sizes. And next I will talk about more on effect sizes. So effect sizes is represented by theta, as I said. So theta k represents the effect size of study k, and then theta k, the one with hat, theta hat k, represents the observed effect size. So if you this is a true effect size, and you would like to approximately measure the true effect size by this theta hat. This is the observed effect size that you will probably measure. that you find it in publications, or when you run your analysis, and that's the effect size that you measure, which is actually that you are aspiring to measure the true effect size. This is your ambition. So the observed effect size is measured from published studies, and it's an estimate of the true effect size. Usually you're trying to estimate the true effect size. And how close this has to be? It's based on some kind of error, and that is called sampling error. And the observed effect size approximates, as I said, the true effect size. And so it should be, this is the true effect size, and this is effect size theta hat that you measure from your studies, and then there is some kind of sampling error. So if your sampling error is very small, so that means your sample size is very big, that means you are approximately measuring the true effect size. If your design is excellent and your sample size is very big, so that means you will be able to at least closely, you may not be able to hit the exact figure, the exact true effect size, but you will be approximately be able to measure the true effect size. So just like theta k, that means the true effect size, this error, the sampling error is unknown. They are unknown, you don't know them, but you try to approximately measure them. Then theta k, you try to approximately measure it by theta k hat, and the sampling error, this one, the sampling error, and you try to measure that by statistical approximation. That's usually the standard deviation of sampling distribution or standard error. So here are some measures of effect size in single group designs and in multiple in control group designs. So in single group designs, you may use mean, proportion, correlation, and in group designs, you may have effect size in group, and actually you may have in group designs, you may have mean differences. It could be standardized mean difference or ANCOVA adjusted mean difference. There are so many different types of mean difference, or within group mean difference, or between group mean difference. And some other measures, like if you deal with categorical outcomes, you may use risk ratios, or risk ratios, hazard ratios, and incidence rate ratios. So this is a very basic introduction for like a start for our next session. So for today, this is the introduction to the meta-analysis section. Next, we will talk about central limit theorem and more about all synthesizing and synthesizing more to the theoretical aspects of how you synthesize and based on our outline. Yeah, so we'll talk more about pooling effect sizes and the like. I hope that gives you a brief introduction about meta-analysis, and I would like to see you next time. Until then, enjoy meta-analysis, and please like and subscribe so that others can be able to see the algorithms will push to other people who are interested in the topic. Thank you.